Welcome to TIBR classes. Today we can discuss about cytoskeleton. The cytoskeleton provides structural support to the cell. Like our skeleton, which provide support to our body, each cell in our body has a separate skeleton which involved in cell motility, its regulation, everything. So cytoskeleton, the cell skeleton is more important. As I said in the previous slide, the cytoskeleton, the main function is providing mechanical support to the cell, maintain the shape of the cell, provides anchorage to the organelles. Organelles have been shifted from one position to another through this the cytoskeletal components. They are dynamic in nature. They can dismantle at one spot and reassemble at another spot so that it can change the cell shape. Cytoskeleton is a network of fibers extending throughout the cytoplasm. You can see the fibers in the electron micrograph. It is a network of fibers which organizes the structures and activities of the cells. Cytoskeleton plays a major role in cell motility. The cytoskeleton interacts with motor proteins like dynein, which is involved in the retrograde movement from plasma membrane. Retrograde movement means the movement of components from plasma membrane to nuclear area, as well as kinesin, which involved in the anterograde movement to the plasma membrane from nucleus to plasma membrane, helping in transport of organelles, proteins, vesicles within the cell. It's important thing you need to remember that dynein is the motor protein involved in retrograde transport. Retrograde transport means from plasma membrane to nucleus, while kinesin involved in anterograde transport from nucleus to plasma membrane. You need to remember these 2 points for your exams. The motor proteins, which I have mentioned in the earlier slide, dynein and kinesin, carry vesicles, organelles to various destinations along the monorails provided by the cytoskeleton. Cytoskeleton acts as a road or monorail through which this motor protein carry the cargo to different parts of the cells. Its evidence is accumulating that cytoskeleton may transmit mechanical signal that rearrange nucleoli and other structure. Nucleoli and other structure rearrangement can be done by mediated using the cytoskeletal rearrangement. Cytoskeletal rearrangement can result in the rearrangement of nucleoli and other structures. This is a representation in which how the motor proteins move through the cytoskeleton. There are 3 main components for cytoskeleton, which includes microtubules, microfilaments, and intermediate filaments. Coming to the components of cytoskeleton, first the microtubules. They are hollow tubes formed by the protein called tubulin. Tubulin can be of alpha tubulin and beta tubulin, where diameter is about 25 nanometer with 50 nanometer lumen inside. Their main function is maintenance of cell shape, cell motility, chromosome movement during cell division, organelle movement, etc. Coming to the second type of component of cytoskeleton include microfilament, or they are also called actin filament. They are composed of the protein actin there are two intervened strands of actin forms the microfilament. Their main function are the maintenance of shape, change in cell shape, muscle contraction, cytoplasmic streaming, cell motility, mainly pseudopodia, main motility by amoeba, as you have seen. It is brought about by this action of microfilaments. Cell division, the cleavage formation. So during cell division, after nuclear division, cytoplasmic division occurs in animal cells by cleavage formation. For cleavage formation, the actin filaments plays an important role. And the third component of the cytoskeleton include intermediate filament. They are fibrous proteins supercoiled into thicker cables. Mainly include one or several different proteins of keratin family, depending on the cell type. Keratin is the protein which forms the intermediate filament. Then main function and maintenance of cell shape, anchorage of nucleus and certain other organelle, and formation of nuclear lamina. Nuclear in the wall of the inside wall of the nucleus called nuclear lamina, in which various important metabolic process occurs. So the formation of nuclear lamina is carried out by the intermediate filaments. Coming to microtubules in detail, the microtubules grow from the centrosome near the nucleus. Centrosome is an organelle which serves as the microtubular organizing center. And in animal cells, the centrosome has a pair of centrioles, each with 9 triplets of microtubule arranged in a ring. And this microtubular structure is made up of the protein tubulin, as I said earlier. During cell division, the centrioles replicate and moves to the opposite poles. The microtubules are the central structural component which supports the cilia and flagella. In cilia and flagella, the microtubules form 9 doublets oriented with 2 additional microtubules, form a wheel-shaped structure. This arrangement is usually called as 9+2 arrangement, wherein each doublet is connected one another by the protein dynein. As both flagella and cilia are structural components of the cell and are maintained by the microtubules, they can be considered as a part of cytoskeleton. So flagella and cilia is also a part of cytoskeleton. They are composed of microtubules. They are arranged in 9+2 arrangement. I will show the 9+2 arrangement in next slide. This is a 9+2 arrangement. You can see the micrograph of a wheel-shaped structure, of which 9 duplex forms the wheel outside, and inside there is also 2 microtubules are seen. So 9+2 arrangement. This is known as the 9+2 arrangement of cilia as well as flagella. Then coming to the microfilaments, they are the thinnest class of cytoskeletal fibers and are solid rods of globular protein called actin. Actin makes the microfilaments. An actin microfilament consists of twisted double chain of actin subunit. Actin filament mainly consists of 2 chains which are twisted each other. The microfilaments are designed to resist tension. With other protein, they form 3-dimensional network just inside the plasma membrane. During muscle contraction, you may be familiar with the actin myosin involved in the muscle contraction. So in muscle cells, thousands of actin filaments are arranged in parallel one another, and the thicker filament, you can see the thicker filament which is composed of the motor protein myosin. As we have said about dynein and kinesin, there is another motor protein called myosin interdigitate with the thinner actin filament. In between the actin filament, we can see the thicker myosin filament. This myosin molecule walk through the actin filament and pull the stacks of actin fiber together and shorten the cell, help in the contraction of the muscle cells. As we have seen in dynein and kinesin transporting molecule, like that the smaller the actin through the actin filament, this myosin, the thicker filament or the myosin, the motor protein, walks through this actin filament so that it pulls the actin filaments together so that it results in the shortening of the cells. This is the mechanism behind the muscle contraction. In contrast to the muscle cells, in other cells, actin myosin aggregates are less organized, but still cause some localized contraction. During cell division, the cytokinesis occur by means of the formation of contracting belt of microfilaments, as well as in amoeboid movement, the pseudopodial movement occurs by the action of these actin filaments. In plants, this actin myosin interaction plays an important role in a process called cytoplasmic streaming. This creates a circular flow of cytoplasm in the cell. This actin myosin interaction results in the streaming of cytoplasm throughout the cells. This speeds the distribution of materials within the cells. That transport of materials within the cells is carried out by cytoplasmic streaming in plant cells. This is mediated by actin myosin interactions. Now the third class, intermediate filament. Intermediate filament are intermediate in size, 8 to 12 nanometer in size. They are specialized to bearing tension. Intermediate filament are built from a diverse class of subunits from 70 different protein. It is not made up of tubulin or actin or specific single type of protein. It is made up of 70 different type of protein polymerized to form these fibers. Intermediate filaments are more permanent fixtures of the cytoskeleton than other two classes. Other two classes help in the movement of the cytoplasm or movement of the organelles, etc. But these intermediate filaments are usually permanent fixtures. They reinforce the cell shape and fix the organelle location. Some of the different intermediate filaments are vimentin. Vimentin is an intermediate filament which is generally present in mesenchymal cells. Keratin is present in mainly epithelial cells. Neurofilaments is present in neural cells. Lamin, which giving structural support to the nuclear envelope. Desmin plays an important role in the structural and mechanical support to muscle cells. Crescentin, which is a protein which is found in bacteria called Colobacter crescentus. They are related to the intermediate filaments of eukaryotic cells, which confirms some evolutionary relationship of the cytoskeletal of bacteria as well as cytoskeletal of humans. The crescentin is also involved in the maintaining the cell shape, such as helical or vibrio form of the bacteria. But the mechanism by which it forms or maintains a cell shape is currently unclear. So it connects us with the prokaryotes. So let us conclude the microtubules, which mainly made up of alpha tubulin and beta tubulin, and microfilaments mainly made up of actin, and intermediate filament which made up of keratin, vimentin, lamin, etc. are very important in maintaining the cell shape, helping in cell motility, helping bacteria in finding its food. Like that, the normal metabolism of cell is usually maintained by the cytoskeletal subunit. So thank you for watching this video. Please subscribe to this channel. We will come with more videos which will help you in preparing competitive exams. Thank you.